Welcome everybody to our annual PhD research meeting and to the final of our 2016 symposium series. The department depends on the research undertaken by our undergraduates, by our PhD students. So our PhD students are really the lifeblood of the research that we do, and you provide the intellectual muscle for what we achieve. So I'm really thrilled to see the lineup of talks that we have today, and I really look forward to hearing from you all what you've been doing over your time here at Imperial. We're very lucky today to be able to combine the PhD symposium with the final lecture of our 2016 symposium series. So as you know, our distinguished seminar series aims to invite to our department distinguished speakers from around the world, have them visit our department, learn about our research and teaching activities, meet with our staff and students and our colleagues in their research groups, and get an idea of the breadth and depth of the research going on in the department. It's also a unique opportunity for us to be informed of the activities of our distinguished speakers. And we're lucky today that our final symposium speaker for 2016 is able to participate in our PhD research symposium. And today's distinguished speaker is Professor Joaquim Cabral from Instituto Superior Técnico in Portugal, so from Lisbon. Professor Cabral obtained his diploma in chemical engineering in, I don't want to embarrass you, but 1976. And his PhD in habilitation, or what we would call in the UK, Doctor of Science in Biochemical Engineering in 1982 for the PhD and 1988 for the habilitation. In 1983, he left Portugal and went to MIT for a postdoctoral period before he returned back to IST, where he joined the faculty as an assistant professor. And he rose through the ranks there to become a full professor in 1992. From 2002 to 2009, he was the head of the Department of Chemical and Biological Engineering at IST, and currently he's a professor of biological engineering in the Department of Bioengineering at IST, which I believe is a newer department which has been set up there. And in fact, he is the founding head of this Department of Bioengineering. And as well as being the head of the Department of Bioengineering, he's the director of the Institute for Bioengineering and Biosciences. In May 2015, he's the first faculty member of IST to be named an IST Distinguished Professor. So I think that, for those of you familiar with IST, you know that that is really a great honor. We've had a number of students from IST pass through this department, and I think we would recognize them as being genuinely excellent students. I think that's a real honor to be the first Distinguished Professor from IST. He's also at IST the director of the doctoral program on bioengineering in collaboration with MIT. And he's been in numerous advisory boards, and he's currently the president of the Exact Sciences and Engineering Scientific Council of the Portuguese Foundation for Science and Technology. He's also the associate editor of Biotechnology and Applied Biochemistry. Professor Cabral is internationally known in the field of biochemical engineering and bioprocess engineering. He's published over 470 research papers. He has an H factor, for those of you that are interested in these things, approaching 50, and he's co-authored 6 books. His present research is focused on stem cell bioprocessing and biomanufacturing, addressing the ex vivo expansion, controlled differentiation, and purification of stem cells, in particular hemopoietic and, Sarkis has written this word to test me out, I know, but I'll say it, mesenchymal stem cells, as well as embryonic and induced pluripotency stem cells for clinical applications. So please join me in warmly welcoming Professor Cabral to give our final 2016 symposium series.
Thank you. Thank you very much. Okay, let's see. Let's see if I... Okay, thank you very much, Professor Andrew Livingstone, for your nice introduction and also for the invitation to be here today. We met already a few years, not so long time ago, a few years ago, when we visited Portugal, and also we have been in committees at Universidade Nova and IST. And also I'd like to thank Professor Stackis Mantalaris also for this invitation and all of support that I got through this. It's for me an honor to be here and to share with you some of our activities, mainly the research activities, as we are here today in the PhD symposium. Of course, the PhD, I will also grade the undergrad students. Of course, they can contribute to that as well, but as you mentioned. But of course, the driving force for a university is through the doctoral students and postdocs. So today, my past is in chemical engineering and in the bioprocess engineering. So I moved from biocatalysis, fermentation, and when I was at MIT with Charlie Cooney and Danny Wang, it was in fermentation, downstream processing. So, but more recently, let's say this century, in fact, in 2000, I moved to apply the same concepts that we have in bioprocess engineering to the stem cell field. So that's why I put this challenging title, I hope, for bioprocess engineering strategies for stem cell-based therapies and regenerative medicine. So the outline of my presentation here today is I will start with the motivation. What is the motivation to be in this field? And the motivation is mainly based in stem cell-based therapies. The current ones that are mainly based on the hematopoietic and mesenchymal stem cell therapies, namely one type of disease. This is the graft versus host disease. So that's, I will focus on that. We have other activities also in cardiac applications, but I will focus in the hematopoietic. And afterwards, due to the limitations that we are facing currently to this translation aspect, I will go through the more research oriented in stem cell process engineering, namely combined bioreactor and microcarrier technologies, both for mesenchymal stem cells as well as for iPS cells, so induced pluripotent stem cells. So hematopoietic cell therapy, which we can say that is has been known for years as bone marrow transplantation. Okay? That is the gold standard of that is a life-saving procedure to fully recover or establish long-term hematopoiesis after the destruction of the hematopoietic system by chemo or radiation therapies. So the first evidence of that, it was in the 60s, after the post-World War II, the radioactive effects on that. So today, the bone marrow transplantations, or if you want, hematopoietic stem cell therapies, have different applications in hematological, immunological, and oncological applications. Namely, for example, the treatment of malign disease like lymphomas, leukemias, hematopoietic stem cell rescue in cancer chemotherapy. This is one important application. Other applications that are not related with oncological, but mainly with hematopoietic disease, blood disorders for children, for example, like the thalassemia and aplastic anemia, metabolic diseases, also for early syndrome, and immunodeficiencies. So these are the gold standard of this hematopoietic stem cell transplantation. However, after bone marrow transplantation, some cases occurred, and the causes of death after allogenic bone marrow transplantation, because you have to have a compatible donor, HLA donor, to the patient, we have relapse that counts for 40%, infection 15%, viruses, fungi, and so on. Related regime toxicities, another 15%. On green, I have here the graft versus host disease. This is a disease that is developed after the bone marrow transplantation when there is the rejection of the transplant, and other causes that is 15%. So in order to treat this, the potential for stem cell engineering and therapy in bone marrow transplantation, I identify here in this cartoon, for example, the regime-related toxicity, as well as GVHD, is accounted. One way to treat that is by mesenchymal stem cell therapies. So this is will focus. I will not go in terms of the infection or allo-specific donor T cells, for example, against cytomegalovirus, EBR virus, and fungi, and others, for example, for relapse, that is the donor lymphocyte infusion, so it's a disease specific. So we move this from the hematopoietic stem cell to mesenchymal stem cell therapy. So mesenchymal stem cell therapy, the aim is intravenous cell therapy with a main objective to restore function of the damage of diseased tissue, avoid host rejection, and reduce inflammation without use of immunosuppressive drugs. So the initial applications of that for mesenchymal stem cell therapy are the direct contact of the differentiation of mesenchymal stem cell lineage, namely for repairing of the potential for osteogenic cells to produce bone tissue or cartilage. This is one example. So this is more oriented to the tissue engineering aspect. But more recently, the current clinical trials focus on this therapy is mainly not to the direct differentiation or engraftment, but through the trophic mechanism, namely the secretion of cytokines, as well as by the exosomes of the mesenchymal cell therapy. So this is one aspect that was introduced 5 years ago by Kaplan in 2011, and this is mainly a cellular therapy. So maybe the future of that is not to use the cells at all for the therapy, but to use the molecules that are produced by this type of cells in order to have the function of this mesenchymal stem cell therapy. So we move mainly, maybe we can foresee that the advance in this area, it will be more a molecular therapy rather than a cellular therapy in the future. So the ex vivo expansion of mesenchymal stem cells for therapy, the clinical doses that are needed are at least 1 million cells per kilo of patient. So usually the bone marrow sample has only, for the donor, has only around 100 to 1,000 primary mesenchymal stem cells, which means that we need expansion, so growing of these cells outside of the body, so is an ex vivo, and in order to get the number of cells that are needed for the particular therapies. So on the left side, we see that some of the applications of that for orthopedic defects, for example, for tissue engineering, many of those are from autologous. So it was from the patient himself or herself. But for cardiovascular, neural, and others, and GVHD is mainly for the allogenic transplantation. It means that by a compatible donor to this type of disease. So here I'd like to share with you one project that we started in 2007 that was supported partially by the MIT Portugal program, but it was mainly done between my university or the School of Engineering, IST, the Portuguese Institute of Oncology in Lisbon, and the Histocompatibility Center of the South, that is where we have the possibility to use the tissues and cells from the donors. So this was supported also by a private group in Portugal, Zemel Saúde. So in this particular case, the driving it was by the Portuguese Institute of Oncology. So we used the clean room facilities of the Histocompatibility Center of the South. So in order to perform all the expansion of the cells in GMP conditions, and in order to get the infusion of the cells to the patient. So by that time, until 2010, we treated 8 clinical cases. This is with Professor Manuel Cassis from the Portuguese Institute of Oncology. 4 of them, in fact, the graft-versus-host disease, 3 acute systems and 1 chronic. I shall say that the 3 acute were received well in terms of the mesenchymal stem cell therapy. The chronic, the results were not so convinced at all. So the other 4 were mainly for children disease, blood diseases like aplastic anemia, Early syndrome, and so this was the most motivation of this practical and cases that we came. However, if we look to these applications from the process point of view, because we are here in a chemical engineering, I'm a chemical engineer, a biochemical engineer too, so we have to look what are the process limitations. So the process limitations are the classic expansion is very time consuming. So it is needed at least 3 to 4 weeks, it means 1 month, to grow the cells, not in a bioreactor system, but on tea flasks. So as a tea flask, you have a planar surface, so mesenchymal stem cells are adherent cells. As you probably know, only the hematopoietic stem cells are the ones that are in suspension in our body, but all the other ones are adherent, so they need a surface to grow, especially if it's ex vivo. So the growth of these cells is on the surface of these tea flasks. And you have to proceed several passages in order to get the higher number of cells. Of course, there is also a risk in terms of genomic defects, and so usually the surgeons or the hematologists don't want to go further than 2 or 3 passages in order to be on the safe side to the patient. Okay? So this is a very time-consuming, is a very expensive process. So clean room facilities, GMP facilities for that, of course, large volume of cultures, and qualified serum batches. I'd like to point out this because it's an important issue. So currently, even today, what is going in terms of the clinical applications, it is to use a medium that is rich in serum. Okay? The serum is mainly from bovine serum, but should be a qualified serum. For example, in Europe there is a group, this Leiden group, that there are several countries where these batches are qualified in order to be sure that are safe to the patients. And for that, the bovine serum should come from countries that have no problems with mad cows disease and so on. So most of these are coming from Australia and New Zealand. Okay, in fact, this qualified. I don't know for as here, but this is one aspect. Another aspect is the limit cell expansion. So we have a limit cell expansion. How many doubling times or doubling cells we can get with this process? So these are the process limitations currently. Another limitation is from the cells themselves. So the cells, mesenchymal stem cells are quite rare. So, for example, for a newborn, we have one of these type of per 10,000. But after that, with the teen and with aging, so there is a strong decrease in number of mesenchymal stem cells available in order that can be used for allogenic applications. So when we needed at least 1 million of these type of cells per kilo, it means that we need a lot. It means that we need millions of cells. Okay, you need usually for an adult is around 160 millions for an adult, because for this type of treatment there is not only 1 infusion but at least 2 infusions. If an adult has 80 kilos, you can see that you needed 160 million cells. So this is the need for the expansion. So what we think about, we have to think as a process engineering is to scale up this expansion, this growth. So from a Petri dish or a tea flask through a spinner flask, it is not, of course, a controlled bioreactor system, it is a stirred system anyway, to a bioreactor system. So in terms of time scale. So the limitation of the static culture, as you know, as you can foresee, is the low productivity. So we have a limited number of cells per surface area, is a non-homogeneous nature, so we have no mixing at all of the medium. It is difficult to monitor and control. There is a lot of time consuming in terms of handling and feeding, harvesting procedure, and absence of flow in this type of culture flasks. So the alternative systems should allow a better homogeneity, a tighter control, and a higher productivity of cells. And so we move to the controlled culture systems in bioreactors. At IST and in collaboration with the different hospitals that we have mainly in the Lisbon area, mesenchymal stem cell sources are that we started with bone marrow, so mesenchymal stem cells that were isolated, for example, after consent, of course, by the medical doctors at the Oncology Institute. So bone marrow is a simple isolation procedure, however, is a very, very invasive one. So the donor has to be submitted to this. In the last 10 years, I should say, umbilical cord is a very interesting mesenchymal stem cells source. So it's available, low isolation rate in the blood, but in the cord matrix, in fact, we can get much more than that. Isolation is quite easy, and so as it is from a newborn, we have much more primitive cells, as I mentioned before, rather than adult stem cells. Adipose tissue has been also another one for aesthetic reasons mainly, but has been also used, and synovial tissue that is much more committed to the cartilage has been also one that we have in collaboration with the orthopedic departments. So in terms of one technology, when we face the cell grow, as engineers we can think how to grow this type of cells. So they can grow on surfaces or they can grow as aggregates. So today here I will share with you what we have been done with microcarrier-based cultures. In fact, we have also looking and working with aggregates in order to get higher number of cells too. But the first idea is that if we need a surface for cells to grow, the easiest one and to have a higher number of cells is to use a microcarrier. So it's a spheric, so it's relatively easy to do. So we have to look to the different aspects, namely the cells, microcarriers, the medium, the bioreactor itself, and the downstream processing. So cells, microcarriers, the cell source, the selection of the microcarriers based on the chemical, physical, and geometrical properties, the coating substrate in order to promote more interactions between the cells and the surface of the microcarrier, because one of the first definitions of mesenchymal stem cells is that they adhere to plastic. So we can use plastic microcarriers, but also we can, for other applications or add different type of cells, we can improve that surface using elements from the extracellular matrix or vitronectin, and depending on the type of cells as well. So medium is an important one. So attachment and the growth factors, shear and protective agents. And with medium, I will look in more in detail in a few slides. The bioreactor parameters, so temperature, dissolved oxygen, pH, steering protocol, configuration, aeration, feeding regime, and of course, the most also important issue is the downstream processing that I will not tackle today here too much, but is an important in order to have the cell harvest protocol, the recovery, as well as the characterization, and most important is the quality of the cells. So our product, when you compare to the bioprocess engineering, the product is the cell itself. So the cell itself should be high quality in order to be safe to be used for applications. So here in this slide, I summarize a few publications that we have, and also the process. Because we started with, of course, before in tea flasks, but after that, in the spinner flask as an intermediate step, using a medium, but we decreased the quantity of the serum in the medium. So in order to adapt, because usually the medium has 10% of serum, so we start to adapt the cultures to lower and lower medium. So one intermediate step, it was to use 2% of medium and using Cultisphere S microcarriers. Cultisphere S microcarriers are microcarriers that are well known for vaccine production and so on, so are based on gelatin. Okay, so gelatin is is a substrate, I should say, that promotes also the interaction between cells and the microcarrier itself. So we moved to the plastic microcarriers afterwards in order to replace Cultisphere S, because Cultisphere has gelatin from animal origin. So we're trying to adapt this as a strategy to remove all the animal components in the medium or in the culture. So one aspect is we replace the type of gelatin microcarriers by plastic microcarriers. And the next step, it was to use a Zenofree medium. So it means that we don't have any animal components in the medium. And of course, after going to the bioreactor system, 1 liter bioreactor, so 800 mils volume. So this was the strategy. I will show you that one of the issues that is important is the risks, as I mentioned, from fetal bovine serum. So is a source of animal proteins as a resume, bacteria, virus, or prions, xenogenic antibodies, and poor definition, and as well as variability batch to batch. So in order to have a more robust system, so we should also go to the Zenofree medium and plastic microcarriers. So we published a few years ago in 2011 this paper in tissue engineering. And this has been done also in collaboration with Life Technologies today as thermoscientific, because they are one, it's not only exclusive, but one of the companies that are developing Zenofree medium. Okay, so Zenofree medium is one aspect that has been under research, and we have been working with them for a certain number of years. So we start with mesenchymal, hematopoietic, and now also with iPS cells. So for example, an expansion even in spinner flask, expansion of these mesenchymal stem cells from different origins, we can say the highest expansion that we get and highest grow, it was with the umbilical cord matrix mesenchymal stem cells. As I mentioned before, those are more primitive cells, so we expect to get higher number of cells as well as higher growth rate. So more committed one, it was the synovial membrane, so it was more committed to the cartilage, for example, differentiation, and so that has also impact and depends, of course, in the medium. So with this, we are able to define Zenofree microcarrier-based culture in spinner flask in that case, and for the expansion of human mesenchymal stem cells. So in bioreactor, it was the next step when we also published in Biotechnical Bioengineering 3 years ago, I suppose, with this work. And one aspect that is the aeration, or if you want, the oxygen that we have in culture. So we perform, before that, I did not show here, but before that, we had another paper in Journal of Cellular Physiology where we proved that it's natural, that under hypoxia, the cells grow better than in normoxia, because this is expected. Because in our bone marrow, for example, the cells are not facing the 20% of air that we are, oxygen, okay? It's much lower than that, it's around 2% or so. So if you control the oxygen in the bioreactor system in order to get closer conditions to the in vivo, so of course we favor naturally the growth of these cells. What happens is mainly, I did not show this here, but I can share with you. This is the lag phase is much shorter, at least 1 or 2 days shorter than in normoxia. And after the cells start growing and have this. So under these conditions, we can see that the best results were obtained when we have in the bioreactor 20 or 90%, it means that is equivalent to the 4% or 2% in the static conditions. So with that, we select these two or this range to be used in the bioreactor system. So under those conditions, we can get, for example, after 8 days, 120 million cells in 1 liter bioreactor system that I present here. This is a picture from our lab. So in one of these, we can get this number of cells. And even if you look to the feed regime as well, we can try to adapt more and more, or so increase the number of cells. And for example, when we have a fed batch system of concentrated feeds of nutrient and growth factors every 2 days that are adding, so we can get up to 160 million cells in total for this. So it means that if we target a clinical expansion of 1 million cells per kilo, so if you have 2 doses, I mean, as that example of a patient of 80 kilos, it means that we can get, we need 160 million cells per patient. So in static conditions, that's what we did before. I can say that took 1 month, so 3 weeks from day 0, the MSC isolation, and after in T flasks under static with 10 T flasks passage 1 and with 50 T flasks passage 2. That means it was handling. It was one of my, both of them are here. One of them are here, the other was in the former picture of this project with the Portuguese Institute of Oncology. And we can see that is handling, a lot of handling by the, and in fact it was a former PhD of mine, Francisco Santos, and Claudio Lobato, that is assistant professor in our department that do it. However, if we use a bioreactor system with this 1 liter, with a volume, the 800 mils, we can do it in 10 days, and we can get the same type of number with lower volume of the reactor. So this is the way how we can see and proceed in that term. So of course this is important to have a type of bioreactor that can be used under GMP conditions. So those are not the ones because due to the mechanical parts, but there is here, of course, a challenge and also an opportunity for bioreactor development in order to achieve higher cell numbers instead of to have the static procedures. Also, we could improve more the number of cells. I mentioned already fed batch, but also another type of reactor in terms of operation is the perfusion bioreactor system. So using a perfusion bioreactor system, we can get almost attained by day 11, 200 million cells in that particular case. So the way means in this procedure or this strategy or approach is to with microcarrier systems, plastic microcarriers, Zenofree media, and bioreactors with fed batch, and that can be also combined with perfusion bioreactor systems. So, of course, one aspect that is important is characterization of the expanded mesenchymal stem cells, and there are standard procedures. And one of them is the multi-lineage differentiation ability, for example, for osteogenics, adipogenesis, and chondrogenesis. So with different type of sources, adipose, bone marrow, and umbilical cord matrix, we can get this multi-lineage differentiability. Hematopoietic supportive capacity as well, so this is one test, and the immunophenotypic analysis that is also important to characterize the type of cells that we get after the reactor systems. So, in summary, I put here some figures that are not, that I did not present all of them, but with a different type of cells as well as medium. So the first one is with 2%, the other one is a Zenofree media with cultisphere, with plastic, spinner flask, and bioreactor systems. We have cell concentrations per mil at a certain day. So for example, with perfusion, that's what we get 4 to 10 to the 5th after 8 days in terms of cells per milliliter of culture. So this is a summary of the the results that we have been obtained, or with most of results that we have obtained in this particular case. So this was the first example that I'd like to share with you, based on mesenchymal stem cells, that has a direct application in terms of therapies. And now, more recently, and this is a summary or remarks. So, and the mainly remark is last one, that we can get these 120 million cells at 7 days, or in the perfusion culture, 200 million cells. So the next example that I'd like also to share, we move, sorry, I'm getting my we move to not directly to regenerative medicine applications, but using human-induced pluripotent stem cells. So as you may know, most of you know, I'm sure, that in 2012 Yamanaka got the Nobel Prize in that field, working with or developing induced pluripotent stem cells, so they can be reprogramming of fibroblasts in order to get a more primitive type of cells, like embryonic-like type. So the main use of that is drug screening and toxicity testing. Disease modeling, a lot is being done with disease modeling, and less you can foresee for cellular therapies, but with the big question marks, and so this is not even for the clinical cases, so trials that has been also approached, but should be with a very careful approach on that. So the idea is also to have a scalable processes for this type of cells, to produce, again, high quantity of cells, and to have a defined culture media that is adaptable or is in the GMP conditions. So we start from beginning to work with Zenofree cultures medium. So not start with the medium that are with the fetal bovine serum, but we start from beginning, also again in collaboration with the Fisher Scientific nowadays. So that we use essential 8 medium. It means that have 8 components. In that case, we have polystyrene beads, so plastic microcarriers, but covered or coated. We did a lot of also interactions between different materials in terms of substrate that promote the adhesion of these embryonic-like cells. So in that case, it was vitronectin, and we published this year a paper on that. So also we started with the Zenofree steroid culture, so in spinner flask. One of the major problems here is the cost of the medium, and also the replacement of this medium during culture. This is an extremely expensive process, so aiming to get that in a bioreactor system like we did before, we have to have a lot of funding or collaboration with these type of companies in order that we can promote or have experiments running at that level. So we developed this scalable Zenofree steroid culture. We started with the static, but after we moved to the spinner flask, and we did a factorial design and the optimal conditions that were verified. It was with 44 RPM in that case, and with 55,000 cells per square centimeter. I present before in terms of grams, but or per milliliter, but it's also important is the surface area. And in that case, we can get 1.4 million cells per milliliter at day 10. So this is where we stand now with this type of culture. One aspect that is important is to promote the growing of cells not only in the original beads or microcarriers, but if at a certain point of the culture we add more microcarriers, this is also valid for mesenchymal stem cells. So we can and we prove that we can have bead-to-bead cell transfer, both in static conditions as well as in spinner flasks. So this is one way, because the cells growing as adherent cells to a surface is one way to improve the number of cells, is to add more beads or more microcarriers as a feed regime, a fed batch, if you want, with the microcarriers. So that has been also been done. And another aspect is the multipassage expansion with confluent beads. So if we have a different passage in the spinner flask, we can get up to a fold increase of 135 after 15 days of culture. So, and we can see here in this cytometry analysis that some of the well-known markers for pluripotency were retained, and the differentiation as well is low. So this is proof that we can also have this multipassage expansion with this bead. So finally, one aspect is to use single-use bioreactors. So that is one aspect that I'd like to also to bring to your attention. We are working since last 2 years with a California company, PBS Biotech, that developed these wheel bioreactors. They have different sizes. We have working with them with 0.1 and 0.5. It means 300 mils working volume for the last one. And we can see that in that particular case, we can get cell density of 125 million cells per milliliter, for example, in this 100 mil. So this is one aspect that is important, that have combining Zenofree conditions, microcarrier technology, and single-use bioreactors. Because I think that this type of single-use bioreactor, not specifically these ones that we have tested, of course. I'm not doing propaganda in the trademark, but the concept of a single-use that is also important to be, and the GMP conditions, I think, is the way to go on. So again, this is qualitative assays in order that, for example, by flow cytometry, OCT4, SOX2, NANOG, and the CAEA4 are retained higher. It means that the cells are pluripotent cells. And again, it's a similar type of table that I summarized before. Now with iPS and neural stem cells, iPS-derived cell that I not present here, and we can see that with plastic we have this 1.25 million cells per mil at day 5, and more recently, I did not show here because this is not published yet and is priority, is to use the one aspect is the downstream processing, how to recover the cells from the beads. So one way is to use dissolvable microcarriers. If you use dissolvable microcarriers, so we can facilitate the downstream processing. So, and of course we can integrate in the system. I did not show here, but it's an interesting point, is to also, because due to time limitation, of course, is to integrate all the process. Expansion, differentiation, for example, in neural stem cells, and after recovering of the final cells. So if you use that one, from the expansion to the differentiation is mainly a question to also to move from one medium to the other. So in terms of commitment of the cells. And after for the the downstream processing, the first step, it means the cell harvesting, is to combine in order that if you can dissolve the microcarriers, of course we get the cells free of the microcarriers. So because nowadays, if you want to disrupt the attachment of the cells to the microcarriers, you have to use enzymatic processes, for example with trypsin or with acutase. But in that way, if you have a microcarrier that can be dissolved after the process, so it will be, means that will be free of these microcarriers. So this is in fact the ones that gave a little bit higher cell concentration, 1.5 million cells at day 4. And we are continuing with that in that. So in terms of bioprocess development, in the main remarks, so we have a scalable Zenofree culture system, the multipassage extension, and as well as the B2B transfer, the use of single-use bioreactor systems that produce, for example, after 6 days, 250 million cells. The cell quality and this methodology, I think it will be an issue to continue and constitute a base for future regenerative medicine applications that may require a high number of these type of cells or cell derivatives. So with that, I'd like to finish and to acknowledge, of course, all stem cell bioengineering lab at IST, people that are here as well, that work in the group and are now is at Imperial, my former students. And thank you very much for your attention and also for the opportunity to be here today with you. Thank you.
Welcome. Thank you very much for covering.
Thank you, Andy.
A lot of material. And
yeah, I tried to share with you.
Also for giving such an insight to that field. I don't know much about it, but I come away from that, I think, knowing more than I did when I went into it.
Thank you.
And I open the floor to any questions. Perhaps I'll start off while everyone else is thinking. I noticed that the mesenchymal cells decrease with age. So I found myself thinking, does that mean if you injected mesenchymal cells to an old person like me, I will become young?
I hope to, for myself.
Yeah. So, do you think we... No. So is that a cause, seriously. Or is it are mesenchymal cells related to aging?
I think that mesenchymal cells are related to aging. I'm not expert, of course, in cell biology to answer you properly, but I think so. But one aspect that we should, if it's for tissue engineering applications, it will be an interesting one. So it means that if I broke a bone, so if I have mesenchymal stem cells with osteogenic properties, so how is the regeneration of the bone? Maybe it's a faster regeneration.
So it happens more rapidly.
Rapidly, yeah.
So that's why children heal more rapidly.
Rapidly, yeah.
Further questions? Brad?
To recover the cells from the umbilical cord, does it have to be done immediately, or can the cord or the cord blood?
Is usually quite. There is a period of time. So, because for example, when we got that, we have a collaboration with obstetrician and also the Faculty of Medicine in Lisbon. So they call, and my group is going there to get the cord and start processing the cord. So it's a few hours.
But the reason I ask is, like, if a cord is taken and frozen now, can it be sort of defrosted in 20 or 30 years when there's a need for stem cell therapies for that particular donor, you know, for the child?
Yeah, I know what you mean. So this is, in fact, an issue that has been in the debate, and which proofs do we have? And we don't have proofs at all at the moment. So there is the potential for that. But as you probably know, and you know, that when we have a cryopreservation process with cells and freeze and thaw and so on, some of the cells are dead anyway. So that's why, for example, when we do the isolation is immediately after. Immediate means 3-4 hours, 6 hours after the birth.
Thank you.
Yeah, welcome.
I have a question for the cell questioning from the question is about To another microcarrier.
Yeah.
Do the microcarriers aggregate during the...
Yeah, there we have, depending on the medium, of course, that you use, there is some tendency or trends for the microcarriers to aggregate and to have a bigger cluster of the cells and microcarriers. This is one of the major drawbacks if you want to have a more homogeneity culture system. Okay. But in certain cases, we are able to get quite nice microcarriers.
Thank you, Professor, for your talk. I have two questions. So one is like, have all this achievement been implemented in the industry, and also have this reduced the overall production costs for the stem cell therapies? Because we know that chemically defined medium, it is much cheaper than those produced by serum ones. Yeah.
Yeah. Your question relates with the industrial or the industrial approach on that. So there are, I think that we are facing a new product, if you want, is a cell product itself. So it's a new way of manufacturing. So it's a new industry, if you want. So several companies and not saying not Lohse and Novartis and other ones who are in that, and others are in that direction in order to get this is as a product and as an industry. Especially the dream of the doctors, I know because they share with me, is that, for example, if they needed one type of cell to be used, to have on shelf that type of cell. So like you have a chemical or a bulk chemical or something like that. So that means mainly for allogenics from other person. Okay. So we have two approaches, and we should look to these processes to a different approach. If it's an autologous one, so there is no ethical issues and so on from the. But for that case, you don't have. Usually, I don't think that anyone would like to collect his own cells and keep it frozen or for future applications. But for autologous, the process mainly is the concept of a scaling out process. Okay, so it means that you have 1 T flask, you have 2, 3, 4, 100 T flasks. Okay, for allogenic, is mainly a scale up approach, in order that, in fact, in that case, you can produce and you have that cell very well qualified, like a monoclonal antibody. And if you go in that direction from a transition from cellular to microtherapy, I'm convinced in the future that is one way to do it. So, as a chemical engineer, you should have also this approach. Yes, please. Yeah.
You mentioned about the currently the cell therapy development is the mainstream, but in a little bit more future, you mentioned the protein therapy.
Yeah, maybe because yeah.
How long does it take?
Well,
what is kind of the breakthrough we need to make?
Yeah, I cannot foresee that. Maybe it's quick, but of course, for example, with mesenchymal stem cell. Now, some of the current research is based on the exosomes and to know and characterize that one. So, because we don't know in detail, unless you did a proteomics analysis and also of all the proteins and transcriptomics and so on, that is also useful. Of course, tools to that in order to identify which one is the growth factor that is needed, okay, for a particular element. So, but hopefully that will not take so long.
Hi there, thanks a lot for your talk. It was very interesting. I processed it through the filter of what I know, which is heterogeneous reactions.
Yeah.
So is there anything from that field that you find useful in terms of insights into determining rate controlling steps and limits to growth?
Yeah. So I think that this field is, I should not say that in terms of infancy of the art, but if we make comparison similarities, what happens with fermentation? Okay, that's we can think about that. there is some particularities that are different. So because sometimes it's quite dangerous to repeat exactly the same approach as a fermentation that we did. But looking to the specificity of the product, it means the cell itself. So we can adapt, and of course monitoring, control, and prediction, I think, is a very important issue to the development, the process development of the field.
Okay, well that's a great place, I think, to bring
Thank you.
the lecture to an end. I can see
Thank
some
you.
hands going up.
Yeah,
Thank
but
you
I'm
for being with
available
us
too.
for the rest of the day for morning tea, so
Yeah.
you can maybe ask me any further questions over coffee. It just remains to me to say thank you very much.
Thank you.
Present a small
Thank
gift
you.
to you. Thank you
Thank
very
you,
much
Andy.
for being
Thank
with
you.
us this morning.
Thank you. Thank you very much.